Hello, and welcome to Group 10's presentation on drug delivery.
In this lab, we're trying to model drug release for a spherical agar polymer drug delivery mechanism. In the pharmaceutical industry, there are strict safety regulations and financial stipulations for drug research and design. Human trials are never the first testing opportunity. So, to the efficacy of drug delivery methods, you can first start with the bench model. Our goals in this experiment were to determine the optimal conditions resulting in the highest fraction of dye transferred from the beads to the water. The variables we decided to investigate were both bead count and stir speed. Additionally, we wanted to calculate the drug release rate for each set of conditions. We used tartrazine dye as a qualitative assay for drug delivery, having it diffuse through the agar polymer beads into a stirred beaker of GO water. Absorbance readings from samples were used to then perform calculations that quantitatively determined the rate of diffusion. As you can see in this graphic, our experimental design is meant to serve as an analog to drug delivery in the human body. The introduction of the tartrazine-filled agar beads to the water beaker simulates the intake of a drug through the human digestive tract. After an extended time interval, the dye will diffuse throughout the entirety of the beaker, similar to how a drug would diffuse out of its capsule and be transported to the rest of the body.
So now moving into the theory part of our experiment was focused on changing parameters such as stir speed or bead count and analyzing how it affected the diffusion rate. The main equation we used was m of t over m of infinity equals k times t raised to the n, where k was a constant we found empirically, t was time, and n was exponential diffusion mechanism. For the geometry of the bead and the literature values, our theoretical n should be around 0.43. Once we were able to calculate the dye fraction by tabulating the ratio between m of t over m of infinity, we were able to then take the natural log of both time that we had collected the data and the f values to make a plot. We then linearized this data and were able to draw a trend line in the form of y equals mx plus b. From this line, we were able to extrapolate empirical values for n and for k for both variables and compare them to the n value found in literature. If our n values match that of literature, it would just mean that our experiment was at true Fickian diffusion, which is just the movement of dye from the concentration gradient and no other mechanisms.
To begin, we first make a 1 liter solution of 0.5 gram per liter tartrazine by adding 0.5 grams of tartrazine into 1 liter of water. Next, we measured out about 3 grams of sodium alginate and 300 milliliters of tartrazine solution we just made. The sodium alginate is added into the solution and stirred vigorously until a smooth, uniform solution is formed. We also need to make a 6% weight solution of calcium chloride. To do this, we mixed 400 grams of calcium chloride dihydrate and mixed it with 500 ml of water. The solution is then stirred until all calcium chloride dihydrate is dissolved. To make the beads, we first poured some calcium chloride solution into a weight boat. Then we loaded some alginate solution into a disposable syringe and slowly dispensed it into the weight boat. As shown here, we counted the number of drops to control the amount of beads we have. After the beads are formed, we poured the remaining solution in a waste beaker with a filter paper on top to capture the beads. The beads are then rinsed with DI water and transferred into a clean weight boat. After the experiment has started, we drew samples using a pipette according to the time intervals specified by the lab instruction. The samples are loaded into a cuvette and inserted into a spectrophotometer to measure their absorbance values. We constructed the calibration curve by measuring the absorbance of known concentrations of tartrazine in the spectrophotometer set at 427 nanometers. The regression line showed a highly linear relationship with an R-squared value of 0.9963. We can use the equation of the regression line to convert between the absorbance and the concentration.
This is our absorbance data over time for the two variables we tested. For the first graph, the stir speed was varied. We tested at 100, 200, and 300 RPMs, keeping a constant bead count of 60 beads. The highest absorbance values were found at a stir rate of 200 RPM. For the second graph, the bead count was varied. We tested with 20, 40, and 60 beads, keeping a constant stir speed of 100 RPM. The highest absorbance values were found at a bead count of 60 beads. Using the calibration curve and the density, the observance values over time were converted into mass values over time. The fraction of the dye release was calculated by dividing the mass at a given time by the mass after an hour. The graphs of the fraction of dye release over time for each variable are shown here. These graphs give us a better idea of how the variables affect the diffusion of the dye. Plotting the natural log of time versus the natural log of the fraction of the dye release shows a strong linear relationship. From this relationship, we can calculate the parameters n and k. The slope of the trend line provides the n value in the diffusion equation, and the y-intercept represents the natural log of the k value. Listed here are the parameters calculated from the trend line on the previous graphs. The optimal conditions for better diffusion can be found by comparing the n values to the value for Fickian diffusion, which is 0.43. In our case, our goal was to increase the n value. As shown, the lowest n value is found with 100 RPM and 20 beads. The highest n value is found with 300 RPMs and 60 beads.
Based off of our data analysis, it is our group's findings that a bead count of 60 at 300 RPM produced the fastest rate of diffusion. Out of all the parameters tested, these are the optimal delivery conditions that we would recommend for our drug release. Additionally, our calculations of the exponential diffusion mechanism value, n, for each experiment all yielded values well below the theoretical 0.43 expected for Fickian diffusion of a spherical polymer gel. This would indicate the diffusion of tartrazine into water occurs via a non-Fickian mechanism. However, can this finding really be considered a probable determination? Should our data necessarily be believed? All of our error bars present on our figures indicated one standard deviation above and below the data point. For most experiments, the standard deviation was relatively small, allowing us to have some confidence in the precision of our data between trials. However, the 60B trial at 200 RPM produced an abnormal deviation from the mean, as well as a trend that didn't match theoretical expectations for diffusion. The data trend appears logical, but is vertically shifted above where it should theoretically lie, between the 100 and 300 RPM curves. Additionally, there are many forms of potential experimental error that could have explained our non-Fickian findings. The correlation between the n value of 0.43 and the diffusion model is made under the assumption that the delivery vessel is a spherical polymer. Our beads may have been oddly shaped and deviated from a perfect sphere. We dropped our beads from the syringe high above the solution rather than within it. This may have led to defects. These beads also may have been improperly dried after they were taken out of their synthesis solution and before they were placed in the beaker. This would have introduced a lot of excess tartrazine into solution before the diffusion could occur. Lastly, there may have been some control issues between trials, as beakers and stir bars of varying sizes were used throughout experiments. This could account for differences in rates of stirring between trials despite being set to the same stir speed.
Is that Cheeto dust?